Welcome to this short presentation on mitosis for Biology 121 Lab. What we're trying to accomplish in this presentation, in fact, with the student performance objectives, the SPOs for this lab, is to not have you become with every aspect of mitosis, because that's going to happen in lecture. Our goal really here in lab is just to have you understand how to identify the different stages of mitosis and then know a couple features of the cell itself. Because our goal here is to really have you spend some more time practicing with the microscope by looking at these slides. And also remember, part of the other SPOs for this lab is to have you be able to identify different cell features. You've had a chance to look at red blood cells, you've had a chance to look at human sperm, however, and cheek cells also, but in those cells you're unable to see chromosomes or any spindle fibers. Those features can only be seen when the cell is actually actively dividing. When the cell is dividing, and its nucleus is replicating itself in a process called mitosis. So the other benefit, the other objective of having you guys be able to identify mitotic cells or different stages of mitotic division is it also allows you to take a look at the change in chromosomes as the cell goes through this nuclear division. So once again, the details of mitosis are really going to happen in lecture. Check your SPOs and find out exactly what features of mitosis you're responsible to know, but I can guarantee you we do want you to know the individual stages of mitosis and be able to identify them. In lecture, you'll talk about the details of, in fact, what's happening in each of these stages. So what we see here in this particular slide is a series of mitotic division stages. You'll see this again at the end of the presentation in just about 15 minutes, and you'll be able to identify all of this. I think you'll be quite surprised at how rapidly you can progress through this. But first of all, let's take a look at the function of mitosis. Once again, an item that you'll discuss in lecture, not required for your SPOs, but a good introduction to why we even care about mitosis. Because remember, mitosis deals with the division of a cell, going from one parent cell dividing into two what we call daughter cells. And what's the function of that? Why do we care? Well, any multiple cellular organism is constantly losing cells. Think of this. Every time you chew, every time you swallow food, you're losing cells off the inside of your cheek, your tongue, your esophagus, as it moves through your intestines, so you have to replace these. And you want to replace them with the exact same type of cell that was removed during, in my example, the chewing, the swallowing process. And so we produce identical cells or clone cells through the process of mitosis. So cell replacement is a very important aspect, a feature, a benefit of mitosis. Also, growth in multicellular organisms. Multicellular organisms, so we're talking now beyond the bacterium, beyond the protozoan, begin their life as a fertilized cell called a zygote, which divides into 2 cells, then into 4, 8, 16, 32, 64, 128, 256, and so on, until you get to, for instance, the adult human, which is about 70 trillion cells. Well, how do we get from one single cell up to 70 trillion cells? Through the process of mitosis. Now, each of those cells are genetically identical. Now, granted, some of those cells may turn into heart cells, others turn into eye cells, etc. But genetically, they're actually all the same, and this happened through mitosis. A third and final function of mitosis is asexual reproduction. The prefix a means without, sex means fusion of gametes, eggs and sperm. So when eggs and sperm come together, we talk about that as being sexual reproduction. But many organisms, plants and animals, are capable of replicating themselves, producing another generation of organisms without requiring the fusion of eggs and sperm. This is called asexual reproduction. A good example might be, for instance, a sea star or a starfish. Sea stars or starfish can actually drop off one of their arms, and then that arm regenerates the missing body. The original adult that dropped off the arm regenerates another arm. We wind up with 2 identical individuals, genetic clones of each other, and they've done this without the fusion of eggs and sperm. Really kind of an interesting way of going about replication. And once again, many different types of organisms are capable of doing this. Let's take a look now at the result of mitosis. The result of mitosis is production of diploid clones. The term diploid, the prefix di means 2, ploid refers to the sets of chromosomes. All the cells in our body that are called somatic cells, which you'll learn about in lecture, have diploid diploid chromosome counts. That means that they have one complete set of chromosomes from mom, the maternal set, and another from dad, the paternal set. So you wind up, in the case of humans, with 46 chromosomes in all cells in our body, all the somatic cells, with the exception of eggs and sperm. And so that means that 23 came from the male, 23 came from the female. So we say those cells are diploid. We abbreviate it as 2N. You'll learn more about cells called haploid cells in lecture. So we have diploid clones. A clone, as I've already mentioned, is genetically identical to the cell from which it mitotically divided. And this is the goal. For instance, if we're replacing cells in our mouth, we don't want to replace them with different types of cells. We want those cells to be replaced with the exact same cells that were destroyed that are being replaced. So this is the goal of mitosis, is to produce genetic clones. Now, if we want diversity in cells or diversity in offspring, another form of cellular replication is required called meiosis, maybe known as meiosis. And you'll take a look at that possibly in lecture. What we see in front of us here is what's called the cell cycle. And you see this in virtually every biology textbook. What it's supposed to be here is a graphical representation of what a cell is doing during its life. Let me put that in a different context. If we were to take a cell, or look at a living cell under a microscope, every day for 100 days and watched it go through its normal metabolic activities, we'd find out that about, depending on what kind of cell we're looking at, 15 to maybe 20% of its time, it's actually involved in mitosis. That's what you see here in this blue area. This blue area right here is mitosis. These 4 pieces of pie, if you will. Down here is an area called cytokinesis. We'll talk about that, also part of mitosis. And then down here, all these other 3 pieces of the pie actually represent something called interphase. Interphase isn't actually part of mitosis. Remember, once again, mitosis just refers to division of the nucleus. So what this graphic is trying to show you here, the cell cycle, is that if we looked at that cell each day for 100 days, then about 15 to 20 days out of that time, it would be actively dividing. The rest of the time, it would be engaged in its normal daily activities, cellular activities. And so you'll talk more about this cell cycle when you get into lecture, but what we're concerned about is this right over here, the pie chart, the light and the dark blue, labeled as mitosis. And that's our goal here today. This represents the same concept here of the cell cycle, but from a different artist perspective. And you can see how they've actually indicated that mitosis is only 10% of the time, because it's variable based upon the age of the cell, where the cell came from in the body, etc. Once again, more time will be spent on that when you get into lecture. This is a roadmap of where we're going here in the next couple slides. These are the different stages of mitosis, beginning on the top left-hand side here with interphase. Interphase is actually not part of mitosis, although unfortunately it's labeled as such here. Interphase is basically the warm-up to mitosis. It's kind of like mitotic foreplay here. It's the precursor to mitosis, because what's going to happen in mitosis is we're going from one parent cell, as we can see here on the upper left-hand side, down here to 2 daughter cells. We begin with 1, we end up with 2. Well, if we're going to have 2 daughter cells, that means we have to have 2 nuclei, we have to have 2 complete sets of chromosomes, we have to have 2 complete sets of organelles. So all this replication of organelles and chromosomes is actually happening here in interphase. So interphase, once again, becomes the precursor to mitosis. We're going to look at slides in just a second and see how to identify each of these stages. Once again, we're not asking you to describe the details of what's happening in each one of these stages. Just be able to identify them. That'll help your conversations in lecture go a lot more smoothly when you actually get into the details of mitosis. So we're going to go from interphase to prophase, to metaphase, to anaphase, to telophase. So that's kind of the roadmap of where we're going. Now what you're going to see in the next series of slides in the top left-hand side is the artist's interpretation, and the bottom right-hand side is the actual photograph taken through the microscope of the very same slides that you're looking at, called white fish blastula. A blastula is a very, very early embryonic stage from a fish called a whitefish, actually from the Great Lakes. And so we're going to take a look at how the characteristics of the cell changes between these different stages. Now when we look at interphase, remember this is actually the phase just before mitosis begins, how do we identify when a cell is an interphase? Well, first of all, this represents here, this purple area represents the nuclear membrane. The brown or pink, whatever color that is, area here represents the cellular membrane. Let's take a look over here. Here's our cellular, also known as the plasma membrane, and here is the nuclear membrane. So we can see the corresponding structures between the illustration and the photograph. Now, if we take a look inside the nucleus, what we have in here is what's labeled as chromatin fibers. Chromatin fibers represent the chromosomes that haven't condensed upon themselves. It's an important concept to understand that we can only see in the light microscope, the compound light microscope, objects that actually cast a shadow. So for instance, take a look at the photograph in the bottom right-hand side. All those dark pink or purple areas are areas that are darker and won't allow light to pass through them, so they cast a shadow. That's the image we get. So in interphase, although there are chromosomes there, if you prefer chromatin, you can't see them because they're loosely coiled and they just aren't dense enough to cast a shadow. So we'll talk about how that changes in the next step in just a second. But what is happening in interphase is each of those individual chromosomes, all 23 pairs, all 46 of them, are going to replicate themselves into 46 pairs or 92 individuals. And at that point, when they replicate themselves, they stay attached to their replicate in something called a sister chromatid. Take a look over here on the right-hand side. Here is, I'll outline with the mouse, here is, in this location here, one chromosome that was a single chromosome at the beginning of interphase. As interphase progressed, it replicated itself. Here we have its identical replicate right down here. But they're still attached, kind of attached at the hip right here, with a structure called a centromere. As long as the chromosomes are attached to their replicate, we call them sister chromatids. And then later on, when they separate from each other, the whole purpose for mitosis, we'll call them chromosomes. So down here in this photograph, we can't actually see any of these because the sister chromatids haven't condensed yet. They won't cast a shadow. Let's take a look over here. Here's an illustration of what's going to happen now in the next few steps. Here's the same illustration we just saw. And let's move this purple guy over here, and different artist, unfortunately. We have one chromosome, and its replicate right here. So these replicated chromosomes we call chromatids, sister chromatids. Fortunately, they're no brother chromatids. They're all sister chromatids. But what we're going to have to do is divide these chromosomes into separate cells through mitosis. So how do we pull them apart? We have these structures right here called microtubules. They're also called spindle fibers, as this artist down here has illustrated. Let's take a look at the actual photograph. Here are some sister chromatids that have been stained blue. And then over here, we have the spindle fibers on this side and spindle fibers here that are actually going to pull apart the sister chromatids and make them into individual chromosomes. This begins here in prophase. Let's take a look at prophase. Prophase, the prefix pro, is short for proto, which means before or first. Phase, it's the first phase. Prophase or protophase is the first phase of mitosis. How do we know that we're in prophase? What are our field marks? Take a look here. See the nuclear membrane is beginning to break up? Look down here, little fragments of the nuclear membrane. Look over here. The sister chromatids, which the artist unfortunately has labeled chromosome, we call them chromatids because look, they're attached right here to their replicate. The sister chromatids are now condensing. They're tightly coiling upon themselves, and as a result, look down here in the photograph. They actually cast a shadow for the first time. So these are all randomly distributed sister chromatids at this point. And that's our best field mark for identifying prophase. We see sister chromatids, and they're randomly distributed, and the nuclear membrane is beginning to break up. In this photograph, we can't see what the artist has illustrated here, which is the formation of spindle fibers and the centrioles. But we'll see the spindle fibers in the next photograph. Centrioles are structures that are only found in animal cells. You don't find them in plant cells. And they're nothing more than the place where the spindle fibers attach their opposite end. Let's see what I mean by that as we get into metaphase here in just a second. But you notice there's another slide here that says late prophase. The reason I'm showing you this slide is that understand the whole process of mitosis is a continuous sequence of events. The cell doesn't stop and go boom from prophase to metaphase to anaphase. It's a continuous process. We've rather arbitrarily decided to call these different stages of progression prophase, metaphase, anaphase, telophase based upon a couple of characteristics occurring in the cell. But the cell doesn't know when it's in late prophase or early metaphase, so understand that. And also understand that on the exam, when we ask you to identify these different stages, we'll give you very obvious features. Okay, we're not going to ask you to identify early prophase, late prophase. Okay, so let's go back to the slide we were just at. On the slide before, we go from interphase to get ready for mitosis, and we go to prophase, and then to metaphase. By the way, we'll go through this a second time briefly. Metaphase. The prefix meta means middle phase. This is the phase where the sister chromatids are in the middle of the cell. We use the same terminology to describe the locations in a cell as we do a globe. There's an equator along the center, just like in a globe, and there are two poles, like a north pole and a south pole. And so when we have all of these sister chromatids lined up along the equator, the spindle fibers have attached to each side, getting ready to pull them apart. This is what we call metaphase. Let's look down here. The best way to identify metaphase is look at this, a single line of chromatids. Here are all the spindle fibers attached to this side and to this side, and it's going to pull apart the sister chromatids into individual chromosomes again. Now, the centrioles although they're illustrated here, and you can see one end of the spindle fiber attaches to the sister chromatid, the other end attaches to the centrioles. The centrioles aren't photographed here because when they made this preparation, they made very thin slices through the cell, and apparently the centrioles wound up on one of the other slices in somebody else's slide. So we go from metaphase to anaphase. The prefix ana means up. This is the phase where the sister chromatids break apart and move up towards the poles. So look what's happened here. This chromosome has separated from its identical match over here. When they were together, we called them sister chromatids, but now that they've pulled apart, we call them chromosomes again. And so what are they doing? They're moving up towards the poles. What's our best field mark? Look down here. One distinct row of chromosomes, another distinct row of chromosomes, and this is the stage we call anaphase. The last phase of mitosis is telophase, or telo if you prefer. Telo or telo means end. This is the end phase. It's called the end phase because the chromosomes have now at the end of their travel. They made it all the way up to the poles, and look what's happening. The nuclear membrane is beginning to reform from the very same fragments that formed the original nuclear membrane. Now, as I've mentioned to you several times, remember that mitosis deals with the division of the nucleus. But in order to make two new daughter cells from the original parent cell, the two new cells need not only to have their own nucleus, they need their own cytoplasm, their own organelles, which was also replicated during interphase. So what do we need to do now? Well, we need to divide those. So another process now begins at the same time mitosis is finishing. This process is called cytokinesis. The prefix cyto means cell. Kinesis means to move. It's the movement of the cellular contents, the organelles, and the cytoplasm. How do we know when we're in cytokinesis? Take a look right here. See this dent in the cell membrane? That dent in the cell membrane is called a cleavage furrow. As soon as the cleavage furrow forms, we know that cytokinesis has begun. And we use that as our arbitrary field mark to say, hey, let's call this phase of mitosis telophase. So telophase and cytokinesis begin at the very same time. Let's come down here and take a look at the actual slide. Here's our cleavage furrow, so we know that the process called cytokinesis has now begun. We use that as our indication of what we're going to call telophase in mitosis. Look at this. You can see the new cell membrane that's going to form between the two new daughter cells. What's the conclusion of this whole process? Here's an actual photograph, or actually it looks like a computer animation more like, of two cells just about ready to pull apart, two daughter cells. Here we have the cleavage furrow, and the membrane is going to form right between here, and so we'll wind up with two distinct cells, like this one and this one. So here we have early telophase, here we have later telophase. And what we wind up with then is two distinct cells. Okay, let's do this. Let's go back now and take a look at this. We'll go in reverse here. We'll go back one more time and review this quickly to help you identify each phase. Interphase, the precursor to mitosis. We're doubling everything. Chromosomes. We're doubling organelles. How do we know we're in interphase? You look at the nuclear membrane and you look inside and you can't see any sister chromatids. They're there, we just can't see them because they haven't condensed. What comes next after interphase? Prophase. Prophase we identify because the nuclear membrane is beginning to break up, as we see over here. Most importantly, we see the sister chromatids in no particular geometric arrangement. They're randomly distributed in the nucleus. As we go into metaphase, look what happens. The spindle fibers attach to the sister chromatids, which are all lined up along the middle. So there's an obvious geometric arrangement now as the spindle fibers attach. What comes after metaphase? Anaphase. Anaphase is distinguished by, look at, two distinct lines of not sister chromatids anymore because they pulled apart. We now call them chromosomes. So look at this, two distinct lines of chromosomes. We know that we're in anaphase. What's our last stage in mitosis? Telophase. Telophase, we know we're in telophase because we have this cleavage furrow formed here, which indicates that another process has begun called cytokinesis. We have the reforming here of the nuclear membrane, and eventually what's going to happen is this new membrane will separate the two cells into two completely different daughter cells. Okay, so that's an overview. Let's do this though. Let's go all the way back up to the top and take a look at these slides we saw before and see if you can identify these now. So let's have you take a look at this bottom left-hand slide here and try and figure out, is this a cell that's in interphase, prophase, metaphase, anaphase, telophase? Okay, do you have it? Your answer should be metaphase. What's the characteristic? All the sister chromatids lined up in a straight row and the spindle fibers attached. Let's take a look over here, on the bottom right-hand side. What stage is this? Do you have it? That's telophase. How do we know that? Here's our cleavage furrow, indicating that cytokinesis has begun. Here's our new membrane forming. Let's take a look over here, top center. What stage is that of mitosis? You have it? This is prophase. How do we know? We can see the sister chromatids, but they're randomly distributed. There's no geometric arrangement to them. Take a look over here. Let's take a look at this cell. Here's the cell membrane. Here's the nuclear membrane. What stage is this? Do you have it? This is interphase. We know that because we can't see any we can't see any sister chromatids. We can't see any chromosomes. Come over here. Take a look at this cell. What stage of mitosis is it in? Metaphase, just like the one we saw on the bottom left. All the sister chromatids are lined up in a straight row. Spindle fibers have attached. Our last cell is this guy down here. What stage is this of mitosis? Do you have it? It's anaphase, because we have two distinct rows of chromosomes. Okay, so that's really the short and sweet part of mitosis. You'll learn more details, of course, when you get into lecture, but if you can identify the different stages of mitosis, you know what the difference between sister chromatids and chromosomes, and you know what spindle fibers are, and you know cytokinesis, you should be all set. Make sure that you verify, though, your student performance objectives to make sure that's all the features you need to know, because we change them from semester to semester. If you have any questions, make sure that you get a hold of me. We'll see you on exam day.